It's safe to say that the loss of humanity's staple food crops is a chilling vision. What if it were possible to create a guidebook belonging to each genus of these food crops, allowing our agronomists and plant breeders to improve food security? Fortunately, there is whole genome sequencing, a tool so useful it facilitates QTL and SNP marker identification, providing insights into phylogeny and much more. Accordingly, it's the basis of our food's recently improved resilience and shelf life. The Solanaceae family provides many of our staple foods: tomato, potato, eggplant, and capsicum, the one with heaps of vitamin C. All of these foods have a sequenced genome except capsicum, a genus that prior to 2014 was in need of a whole genome sequence, as the mechanisms controlling fruit size, shape, and yield were largely unknown. However, what was known was the mysterious large size of the capsicum genome, a genome much larger than its sister taxa of tomato and potato. In 2014, a group of researchers set out to sequence the genome of capsicum. The problem was, however, that there are about 35 species of capsicum. To circumvent this issue, the researchers examined 2 capsicum species, each at polarizing ends of the genus: a well-known domesticated capsicum and its wild progenitor. So how did the researchers conduct a whole genome sequence? This was done through a process called de novo assembly and shotgun sequencing. The plants were frozen for DNA extraction. Once extracted, the DNA was randomly cut into many tiny sequences, all of which were paired and read and organized into contigs according to overlap. These contigs were then scaffolded. A high-resolution genetic map of capsicum was then created to orient these scaffolds using complementary SNP markers. This allowed for the scaffolds to be anchored into 12 chromosomes. Through the analysis of expressed sequence tags and bacterial artificial chromosomes, the quality of the genomic assembly was assessed, and in conjunction with the validation of chloroplast insertions into the capsicum genome, several genome libraries for each species were able to be sequenced with high statistical confidence. Next came the gene annotation. In order to annotate the capsicum genes, they were first predicted by using a statistical model that examined exon-intron structure between the genome libraries of the domestic species. Predicted genes were mapped for homologous sequences in a variety of plant species and cross-referenced with RNA sequence evidence obtained through the analysis of protein expression in all tissue types collected throughout all life stages. Thanks to the work of many past geneticists, many of the capsicum genes were identified for functionality by their homologs in several databases. This newly annotated genome helped annotate the wild capsicum genome through a similar procedure. Genes with low levels of overlap and confidence were rejected, leaving capsicum with over 34,000 genes. Once annotated, these genes were run through an orthologous protein sequence algorithm, providing insight into the evolution and functional divergence of the gene families in capsicum contrasted with several other species. This allowed for the construction of a phylogenetic tree demonstrating that capsicum had begun to speciate about 36 million years ago. Although capsicum had left tomato and potato long ago, why was the genome so large? This was found to be the result of repetitive elements transposing themselves throughout the genome. These transposable elements comprised greater than 81% of the capsicum genome. LTR retrotransposons were responsible for most of these transposable elements, and these move via an mRNA intermediate that undergoes reverse transcription into double-stranded DNA, which is then inserted elsewhere into the genome. This copying and pasting of long terminal repeats accounts for the relatively large size of the capsicum genome. Through the cross-reference of LTR fragments in tomato and potato, the insertion times of these LTR retrotransposons were dated, revealing a huge increase in LTR expansion about 300,000 years ago, which is relatively recent for having diverged about 36 million years ago. While this is interesting news for plant phylogenetics, what does this mean for our farmers and agronomists? Fortunately, the discrimination between the genes of domestic and wild capsicum facilitated the identification of genes associated with pathogen resilience, ripening, germination, and so on. This, in conjunction with the comparative analysis of annotated genes between capsicum and other Solanaceae species with a whole sequenced genome, allowed for the identification of genes crucial to the genetic improvement of capsicum.